Good evening, all. I'd like to introduce welcome you to our March mini conference from WAS. We have an evening with our beetle enemies, as our first speaker is identifying the small hive beetle. Lewis is originally from England. You'll catch the accent, I think, pretty soon, by way of California. He has got his PhD at University of Berkeley, California, Berkeley. And he is co-director, I guess co-director of the University of Georgia Bee Lab. He is going to talk to us again this evening, as you can see from his title, Defending Bees from Their Beetle Enemy. Dr. Bartlett, the floor is all yours, and good evening and welcome.
All right, good evening, everyone. Thank you, Dewey, for that introduction. I do miss the West Coast quite a lot. As you mentioned, I was at UC Berkeley for quite a stint of my career, where beetles aren't the same problem that they are here in the Southeast. And actually, a lot of the comparison between my beekeeping experience back in England, where I grew up and where I first learned to beekeep more than 12 years ago now, and then on the West Coast in comparison to here, has really underpinned my interest in trying to get small hive beetles under control, because I didn't really appreciate just how much of an issue they were in parts of the US, and continue to be not only a big problem for us, but an increasingly expansive problem elsewhere in the US, with more and more phone calls my way every late summer from the Great Lakes region and even up into New England of people worrying that their beetles are getting out of control. So we're going to focus on small hive beetle controls today. Some of it's going to be somewhat more abstract science work that's going on in the background of my lab that hopefully at one point will be more applied, but also points a little bit towards how just keeping healthy bees always helps us defend against these sorts of problems. And then we'll talk a little bit more about some of the more acute short-term controls that I'm working on at the moment. And will hopefully be getting into beekeepers' hands in the next couple of years, depending on how working with the companies and with federal regulatory agencies go. So for those of you who might be unfamiliar with what a small hive beetle looks like up close, this is a macro shot of one hiding in some worker comb here, taken by Alex Wild, who is curator of entomology at UT Austin down in Texas. I always say that Alex can do my wedding photographs should I ever get married, because if he's capable of making a small hive beetle look this photogenic, I imagine he can do wonders with people. But this is the adult form of the beetle. Here it is hiding in some honeycomb, just getting ready to cause mischief, to cause trouble for beekeepers who overlap with its major range. So it arrived in the US from Africa in about 1998 via Florida. Most of our invasive problems here in the US when it comes to beekeeping arrive either via Florida or via Georgia or via South Carolina. That tends to be the hot zone for new problem pests arriving. That was true of wax moths. It was true of small hive beetles. It's true of Varroa. And more recently on my patch, it's true of yellow-legged hornets as well. So we're really kind of on the front lines here in the Southeast when it comes to learning to deal with these new problem pests and parasites. With it being native to Africa, the subspecies that we find in Africa of honeybees, the lineage native to most of the African continent, especially sub-Saharan Africa, so Scutellata and other similar subspecies, tend to be pretty good at combating and otherwise resisting infestation from this beetle. Here in the US, most of our managed honeybee stock is derived from European subspecies, with only small areas seeing a lot of those Africanized Scutellata-based genetics, meaning that our colonies tend to be quite vulnerable to a pest that they have no meaningful evolutionary history with. So it's quite troublesome when it does get into our colonies. The life cycle of these beetles kind of underpins some of the current control efforts, which is why I like to spend some time on it. So adults enter colonies and tend to hang out in those colonies, and they're what are first noticed by beekeepers. The adults themselves are minor predators of maybe eggs and a few larvae here and there. But the adult beetles aren't major issues for the colony. Rather, it's when they lay their eggs and those eggs hatch into larvae, it's the larvae that do the majority of damage to the colony. So those larvae feed on pollen and honey, eggs or young brood if they can get to them, and they're burrowing through the comb, through the body of the hive, and through the honeycomb begins to be destructive. They introduce a mucosal slime to honey that leads to fermentation. The smell of that fermentation attracts more beetles, and that slime is also how they defend themselves. So they tend to be cocooned or otherwise shielded in this thick, slimy mucus that entraps adult honeybees that might come over to try and remove those larvae once they reach a critical number and a critical size. Like all beetles, they can only pupate in the soil. That's one of the unifying rules of all beetles, is that their larvae can only develop as pupa in the ground. And so they have to leave the hive in order to undertake that final developmental step of pupation before they become adults. That's important because it's one of the reasons that there's such a problem here in the southeast, and also one of the points at which we might intervene is to try to interrupt that pupation step that occurs outside the hive body. Just to get a sense of how grotesque these things can be when uncontrolled, here's a couple of images that I'm going to scroll through of call-outs that I've had here in North Georgia in the last couple of years. This incident was at a training apiary at one of our local farms. The beekeeper managing these colonies had thrown his back out late June and hadn't been able to get into the hives for about 3 weeks by this point. More importantly, he hadn't been able to take his spring honey from the supers. So these hives were taller than we might otherwise keep them during the summer when our population drops a bit during the dearth. And that excess of unguarded honey created a perfect breeding ground for small hive beetles. So this is the inner cover here visible that I've popped off the top, and you can just see piles of these late instar, nearly fully developed small hive beetle larvae, as well as a lot of dead bees that have succumbed to the mucus. It suffocates them. The entrance of some of these colonies looks like this, where you have this seeping, rotting, fermenting mass of slime and mucus and honey flowing out of the front of the colony. And the smell of this fermenting honey will attract many, many thousands of beetles from the local area. They can fly up to upwards of 30 miles on a good day, and they're phenomenally efficient at sniffing out this kind of vulnerable, unguarded colony. You can see here as well many of the worker bees that have become trapped in this slime and have succumbed to it, unable to reach the larvae that are causing the problem. So as I mentioned, these small hive beetles have this critical pupation step that occurs outside the colony underground in the soil. That means that the weather patterns, the climate, as well as the soil chemistry, the drainage of the local area has a big impact on how easily these larvae survive to be adult beetles, but also their generation time, whereby warm, wet soils allow this pupation step to happen very quickly and allow beetle numbers to build up much more rapidly through the year. And this is a dew point map of the US. Showing essentially a combination of temperature and humidity. And while this is a climate graph of the US, I always say that this could be very easily reinterpreted as a small hive beetle map, with the darkest reds here corresponding to where we see beetles being a problem year round, and then moving up into the Great Lakes and kind of Indiana, Illinois region, where we're increasingly seeing during warm, wet summers big problems with beetles late in the season, and the southeast and the Gulf tends to be really the hot spot for where we see big beetle problems currently. That will likely change as weather patterns continue to alter and we see warmer, longer, wetter summers in parts of the interior US as well. So to summarize some of the risks that lead to these bad small hive beetle outbreaks, the bee breed is really important. Basically, all our US stock that we want to keep at least is vulnerable. Spacious hives and weak colonies. And I know we talk a lot about the idea of a spacious hive being a bad thing in beekeeping. What I often see my newer beekeepers in my patch make the mistake of doing is not realizing that a spacious hive doesn't necessarily mean a depopulated hive. It can mean beekeepers who are a little bit too nervous about taking their spring honey off the top of the colony. So if you're coming out of spring with kind of 3, 4, maybe 5 supers worth of honey on the top of a colony here in Georgia, once we get into the summer and the population maybe shrinks a tiny bit or at least holds steady, that is a lot of space you're asking those bees to guard. And that's where we tend to see the really bad beetle outbreaks, is actually where the colony isn't particularly weak. It's just that someone has left far too many supers on top of the colony for the bees to be able to keep the beetles out. Weather, as I've already talked about, is a really honest predictor of where beetles are going to be a problem. Warm, heavy rainfall tends to be a precursor to big beetle outbreaks. The soil is soft, it's easy for them to burrow into, and it's incubating. I can basically set my watch to 10 to 12 days after we get a tropical storm hit Georgia, because that's about how long it takes before I start getting calls of people reporting biblical plague levels of small hive beetles. So if we get a kind of tropical weather system come through, that always leads to this huge excess of beetles because it's made the soil much more permissive to their growth and reproduction. The final cause that we see sometimes leading to big outbreaks of beetles in a hive is if people are too heavy-handed with protein patties. And this is what actually got me really interested in beetles from a scientific perspective, is I've been working on how we might improve supplemental feeding of colonies, particularly early in the spring here in Georgia, where a package and nuke producing industry principally. And so feeding excess protein in the early season can allow for an extra half a package to a package out of an operation or an apiary, and that can make all the difference. But we can't really feed excess protein or these big protein patties because they become perfect beetle nurseries. That protein patty is just as good, if not better, for raising beetles compared to bees. And once the larvae are inside it, they're protected from the honeybees, and it becomes a perfect breeding ground for them. And so my interest in treating small hive beetles is actually all centered about our inability here in the Southeast to be able to furnish our colonies with these protein substitutes or these pollen patties. The current slate of treatments is, in my opinion, mostly inadequate. You'll see traps, often with vegetable oil in there, which do work for the adult beetles, but they're kind of difficult to maneuver sometimes, and they're not perfect by any stretch of the imagination. Reducing and strengthening hives is kind of our cure-all in beekeeping, so that one goes without saying. And we do see a lot of these nematodes, these entomopathogenic nematodes applied to the soil to intercept those pupa where they will break into the developing beetle and kill it while it's in that vulnerable soil step. This works reasonably well. It relies on your first outbreak of beetles being minor enough that it doesn't kill any colonies, which isn't always the case, particularly in these big outbreaks where we're seeing entire honey supers infested or where large protein patties have been used to rear hundreds of larvae. And if beekeepers aren't raising these nematodes themselves, they can sometimes be vulnerable to being sold kind of dead batches of worms. I often tell my beekeepers that it's worth investing a couple of $100 as a club in a microscope to check that the worms are alive and kicking if they're being bought, or at least just to lie and say you have access to one if you're ordering these, to make sure the supplier doesn't risk sending you a dead batch. One of the reasons I'm so obsessed really about getting these pollen patties into colonies in Georgia is that one of our few defenses against other infectious agents is manipulating the nutritional status of bees. So I'm not trained as an entomologist. I'm trained in infectious disease biology and parasitology, and all my interest in bees came from using them as a study system to understand how viral infection, bacterial infection, and such works. And I've ended up very gladly specializing on honeybees entirely in that vein. But one of the few defenses, for instance, we have against viruses is in ensuring that our bees are well provisioned, not only with sugar, with nectar and honey, but also with protein. And so as an infectious disease researcher, one thing my beekeepers often ask me is what can I do to defend my bees against viruses beyond just controlling for varroa? And one of the few leads we have comes from a lot of work that came out of Amy Toth's lab at Iowa State when her postdoc Adam Dolezal was there, whereby they studied the presence and absence of pollen when doing these controlled virus infections with excess sugar in this case. So the bees aren't starving, it's just whether or not pollen's present. And it drastically improves honeybee survival in the face of acute viral infection if they're furnished with substantial and adequate pollen or similar protein. So this has been one of the few kind of reliable things I've recommended to beekeepers who are particularly concerned about these diseases. We don't have good antivirals for people. We certainly don't have good antivirals for bees. But here in the Southeast, I'm not really able to suggest mass feeding of pollen patties or protein patties as we see elsewhere, such as this image here, which may be very good at preventing viral outbreaks, but would instantaneously lead to a small hive beetle outbreak, much as we see here. I do see we've got one question, so I'm just going to answer that in case it is useful. So two questions, sorry. So Swiffer dryer sheets work reasonably well. That was one question. They're good at catching the adults. No better, in my opinion, than the kind of beetle blaster traps. The bees can sometimes get a bit caught in the Swiffer sheets and they tend to pull them apart. Here in Georgia, our bees are very, very heavy propolisers, and we're mostly a pine timber region, especially here in the north. And so that pine sap, once it starts getting mixed in with the Swiffer sheets, I find unbearably annoying. But they do work, I'd say, on par with the beetle traps themselves. And then with the question about how far the beetle larvae will move leaving the hive to get into the soil, ideally they'll bury as soon as they get into the soil, particularly if the soil is covered with adequate vegetation. If they find themselves on hot concrete or a tarp, they might desiccate or otherwise be predated upon while trying to reach the soil. But anyone who's maintained these in a laboratory will know that they can make it tens to hundreds of meters down the corridor and begin invading your colleagues' offices before they give up. So they can certainly march quite a far distance. So unless you're willing to put your bees kind of in a large asphalted lot, it doesn't really do much. One thing we do have here is an abundance of fire ants that are really preventative in allowing these tasty grubs, as shown here, to reach the soil. But that still won't prevent them infesting supers of honey that are unguarded or infesting protein patties or pollen patties. Okay. So there's insufficient control methods, in my opinion, at the moment. Partly this comes down to the fact that beetles and bees are very closely related, and so there's very few chemicals we can use that are safe for bees but dangerous for beetles. They're also a neighborhood problem. Like I mentioned, they can fly upwards of 30 miles on a good day, and they last a good long while. They can live for up to months, as far as we can tell, meaning that controlling them goes beyond just what happens in your own bee yard. feral colonies, abandoned apiaries can be sources of beetles that are going to come and find your bees. So one aspect of honeybee health that I work on, which is proven promising, that we don't have any advice based on yet, but I like to mention it just to introduce where we're going with some of this, is to really focus on the evolved mechanisms of immunity that honeybees already exploit to see if we can somehow improve their own defensive capabilities against things like beetles. One of these that my new lab group, now that I'm co-directing the Bee Lab here, has been focusing on is the use of hydrogen peroxide in the colony. So this is something that the bees invest in socially. They produce enzymes that generate hydrogen peroxide in the colony and in honey. And for a long time, this was understood to be antimicrobial. What we didn't know was whether it also had a role in defending the colony against bigger threats. So hydrogen peroxide, the same stuff that you'd buy at Walgreens or CVS or wherever else, is present in honey, and the bees intentionally exude an enzyme to generate it in the honey, and this has some defensive capabilities. There are certain times of the year when beetles are more common. It tends to be the same sorts of timescales that Varroa work on. Freezers during the winter tend to kill them off, and then the population builds up over the summer, with the largest numbers typically manifesting in kind of September for us. So hydrogen peroxide, as is found in the colony, tends to be very reactive. We use it to disinfect surfaces. It's used medically and has been for a long time. The 3% hydrogen peroxide that one would buy from the shelf will cause vomiting, even in really quite low doses to animals. Veterinarians will sometimes recommend this if you have a cat or a dog at home that has consumed something it shouldn't have, and there's no veterinarians kind of close by. If you look at the label for what you'd buy in the store, you're actually supposed to dilute it down to 1.5% before even just using it as a mouth rinse. Bees, it turns out, have safe doses far, far higher than we do. And so one of the early pieces of work I did while becoming familiar with this was to just find out how toxic hydrogen peroxide is to bees. And as we can see from these dose response curves here, looking at different colonies as denoted by the different colors here, we really have to get up towards kind of 8 to 10% hydrogen peroxide by volume in sugar solution before we start seeing large, meaningful mortality events in bees. So bees can consume hydrogen peroxide at strengths that would be quite dangerous, actually, to you and I, with that 3% being even then too strong for us to consume. And it's actually quite hard to get hydrogen peroxide into a sugar solution above this 10% point, because even a laboratory grade typically only runs between 30, maybe up to 60% at best. And then you have to dilute down the sugar solution. So bees are extremely tolerant of hydrogen peroxide, beyond what we would even really want to touch. And we don't fully know how they're coping with this. We don't know what adaptations are present. And we did some choice experiments. So here's an example of us feeding some honeybees some hydrogen peroxide solution in this modified syringe here. You can see that despite this potentially being quite toxic to you and I, that she's quite eager to take a sip of the sugar solution here. So we did some choice experiments to see if bees prefer sugar solution with added hydrogen peroxide. Given that they use it themselves, maybe they'd opt for this. And if they do, perhaps we can start feeding it to them as a way to bolster their defenses against things like parasites and pests. These are all what I'd call open questions currently. What levels do they tolerate? What alters this? Turns out bees, as typical in this sphere, and from speaking to other people that have worked on this topic, don't behave as one would predict. And what we actually see in panel A here, which is the better tested version of panel B, is when we mix hydrogen peroxide into sugar solutions, whether that's with glucose or with sucrose, The hydrogen peroxide actually reacts off with the glucose and creates some other molecules that we think the bees can detect in the sucrose. It remains there. But 50 micrograms per milliliter is about in line with what we'd expect of most honey. Not all, some gets quite a bit higher than that. And what we actually see is bees avoid it. So if we're giving bees the choice of sugar solution with no hydrogen peroxide versus sugar solution with an amount of hydrogen peroxide in that kind of looks like what one would find in honey, they actually avoid the one that's spiked with hydrogen peroxide, which we found a bit puzzling and has meant that we've held off so far, really leaning into this as something we want to put inside the colony. Speaking to Mike Simone Finstrom and Margarita Lopez Uribe and a couple of other colleagues who are 5 to 10 years senior to me who have also worked on this, they've also found typically the opposite results to what they expected when working on hydrogen peroxide in bees. So I'm not alone in this, but it maybe explains why not many professors have yet led a significant research agenda examining it. We think that even though they're capable of tolerating it, maybe there's some sublethal cost to them consuming it, and so they avoid it. Another suggestion is that maybe they consider that kind of vintage, high investment honey that they only will use kind of when they have to. What we do know is that hydrogen peroxide has a role in killing small hive beetles. So this was some undergraduate work tracking the survival of small hive beetles at different concentrations of hydrogen peroxide when that's mixed into the food that we're raising them on in the lab. And the most noteworthy one here is that if we remove the hydrogen peroxide through an enzymatic treatment, the small hive beetles tend to survive longer. So even just the amount of hydrogen peroxide that is in honey naturally, between the 40 to maybe 750 at the highest range here, is capable of inhibiting small hive beetles' survival. So beyond just protecting the colony against diseases, the hydrogen peroxide in stored honey products is also defensive against larger enemies like beetles, who we presume don't have quite the same tolerance or defensive capability as honeybees when it comes to consuming the hydrogen peroxide. Excuse me. So we know that honeybees use hydrogen peroxide to defend themselves. They're more tolerant of it than small hive beetles. And while we might not want to feed it to them directly, coming back towards this idea of how do we equip honeybees with the nutritional status they need to defend themselves, another experiment that was done as part of this series was to look at whether feeding sugar syrups, especially during times of starvation, so when there's not much for the bees to forage on, whether that negatively or positively impacts this defense against diseases and pests in the colony. So we'll just start on panel A here. Panel B is the same data, just split up. So we've got the same thing plotted twice, but here in panel A, in the blue labeled 0, we have colonies that received no additional feeding during the summer months here in Georgia, where there's a dearth of things blooming. So there's a span of a few months from typically mid-May, late May to August here in Georgia, where the bees really don't have much to eat, and typically beekeepers will feed during that time. The blue 0 labeled box here represents colonies that were given no additional feed at all. The green one labeled C were fed corn syrup, so high fructose corn syrup. And then the ones labeled S in orange were fed sucrose, so sugar syrup, so table sugar. And what we can see is that the sugar-fed bees tended to be able to defend themselves via this hydrogen peroxide mechanism better than the bees that were experiencing starvation. But the bees fed high fructose corn syrup were basically incapable of generating meaningful amounts of hydrogen peroxide in that stored syrup. And then here on the B panel is the same data, but split into brood frames, which is the B, or honey frames, which is the H. And an interesting feature of this to me biologically is that particularly when they're producing meaningful amounts of hydrogen peroxide, we see significantly higher levels of hydrogen peroxide in honey that is stored on brood frames, suggesting, at least in this early stage to me, that they're tailoring their investment in order to better protect their larvae, who rely on this hydrogen peroxide being mixed into their brood food as part of the honey to protect them against diseases. So kind of an interesting side effect there. But we do know from this work that feeding sucrose solutions, so normal sugar at least, tends to be not only good in that it stops the bees from starving, which the high fructose syrup also does, but that we see higher hydrogen peroxide levels than not feeding at all. And this comes back to my kind of initial idea that I spoke about, that what we're doing in terms of nutritional management for our bees really matters for how they defend themselves against these infectious agents and these pests, whether that's pollen or protein patties, whether it's nectar or sugar solution. We see a direct result on the bee's ability to either communally or even individually fight off infection or fight off parasites. And just to reiterate there, that there tends to be this bias towards them investing more heavily in the brood frames to defend their young. They have kind of mixed results for feeding high fructose corn syrup. It's still a great nutritional energy source for the bees, but it does seem to somehow inhibit immunity for a while. We thought that this was because the high fructose element in high fructose corn syrup interferes with the enzyme that generates hydrogen peroxide. So we mixed up a whole bunch of like weird sugar mixtures in the lab. We had real high fructose corn syrup. We had what we call mock high fructose corn syrup. We had just glucose. We had glucose plus fructose. And what we see is that there's nothing actually that we can pick up that's to do with these fructose glucose ratios. Instead, we think there's some mystery molecule, some trace product of the high fructose corn syrup production that interferes with the mechanism the bees use to generate this hydrogen peroxide. And I am not pursuing what that is because there's no chance that it's ever going to change. And that kind of chemistry is well beyond an innocent bee scientist at this stage of his career. But we do think there's something weird in corn syrup that prevents this immune mechanism working as it should. So once again, as is often the case with the hydrogen peroxide in bees, what we expected to find was not the case. Just as an aside, because this is one of my favorite kind of preemptive answers to questions, we were worried that pasteurizing honey or heating it during extraction would reduce hydrogen peroxide levels in honey. So I sent an undergrad to this who was doing his work in microbiology. And he did lots of beautiful assays of different honey that was heated under different regimes. The purple here corresponds to high hydrogen peroxide levels. And once again, we found exactly the opposite, where it turns out you can heat honey as much as you want, upwards of 80 degrees C. And the glucose oxidase, which generates the hydrogen peroxide, remains intact. We think this is because honey is so thick and viscous that it physically holds the enzyme, the machinery in place, and prevents it being heat damaged. So while we know that hydrogen peroxide is important in honey, in terms of our consumption of it and in terms of using it for, say, wound care, like in MediHoney, that isn't compromised by heating, and how we extract honey doesn't in any way affect this antimicrobial element. Okay, back to small hive beetles. So hydrogen peroxide kills small hive beetles, but the bees avoid it. They tend to use it to protect their young. I don't have a great message for what this means right now, other than feeding your bees when they're hungry tends to be good for their immunity. And as is often the case when we're looking at pests and parasites in hives, if your pillars of beekeeping, and that is a good, strong queen, good nutrition, and a good foraging environment with not too much pesticides in, that will always be your number one defense against things like beetles. Anything else we do doesn't work if your kind of basics of beekeeping are out of sorts. That's not to say we can't still pursue acute controls. So I'm going to finish off now by talking about what I've been trying to develop, which was initially funded by NAPSI. I need to update this slide, apologies. Is now funded by Project APISEM and the National Honey Board, where we're coming up with a chemical control for small hive beetles. And this was work inspired by Dan Potter when he was still at University of Kentucky. He's since retired, and he was working on turf grass management. So we can see here damage caused by lawn grubs, these beetle larvae that exist in turf grass and tend to eat the roots and kill turf as a result. And traditionally, this was managed by using broad spectrum insecticides, so imidacloprid, clothianidin, pyrethrins, and similar. Dan was working for some time on switching over to a newer type of pesticide called chlorantraniliprole, which is an anthranilic diamide. These only came onto market, I think, in 2007, if not slightly later. And he was working mostly on protecting bumblebees from these pesticides used to prevent grass death. And he presented this phenomenal work showing that with clothianidin being used in lawns, you see no queen production in bumblebee colonies at all, which is their main measure of fitness. But that chlorantraniliprole didn't seem to affect this at all compared to control colonies. If we look at colony weight gain in bumblebees over a span of 6 weeks, we can see that clothianidin is very damaging to bumblebee colony development, but that chlorantraniliprole seems to have no effect compared to control colonies. And so walking out of this seminar, I had a couple of pieces of information. One, that this chlorantraniliprole is safe for bumblebees, which are not far physiologically from honeybees at all, and it's perfectly good at killing beetle larvae in turf grass. And so I asked the question as to whether or not we can also kill small hive beetle larvae and it's safe for honeybees. So I went to the location where all good science is born, which is the 24-hour Walmart Supercenter, armed with a P card and a dream, and I bought a sack of this Scott's Grubex. I did some what I call cowboy chemistry in the lab to extract chlorantraniliprole from it, which is only a very, very low dose in this off-the-shelf formulation, and I sprayed it on some small hive beetle larvae, and it seemed to work at killing them. So I got some money to see what we needed to prevent small hive beetles growing in mixtures containing this, what would kill small hive beetles, and see that it's safe for bees. And this is work we've been doing now over the last couple of years. So this looks a lot like raising small hive beetles en masse. We can see some protein patty in a jar here where I've put maybe 30 or so small hive beetle adults. They quickly get to it and produce babies who then create this slimy mixture where they're eagerly feeding on the protein patty. And we simply introduce the chlorantraniliprole into higher and higher doses until we saw, firstly, no successful growth of any larvae, and then secondly, death of the small hive beetle adults. To make sure that we had a kind of safety measure for honeybees and a treatment dose. And the curves here show the difference in sensitivity. This is a log scale, so each of these numbers is a large jump, a tenfold jump, showing that even at the adult stage, the small hive beetles are far, far more sensitive to this chemical than honeybees are. It's actually really hard to even get an LD50 for honeybees with this chemical. It's difficult to get them to feed on enough of it to see large amounts of death in the honeybees. We placed some test patties in colonies outside. These were left for only 4 days in colonies in Georgia before my technician made me get them out of his nukes because he was worried they were going to become overrun with beetles. And our control pollen patties, which had no pesticide in, were in every instance infested with small hive beetles. And when we incubated them, we got thousands and thousands of larvae out of them, whereas our treated pollen patties remained entirely beetle-free in every case. We've hopefully patented this, which is the first step. Grants for large field trials are either in draft or have actually already been awarded, and that work was started last year. And I can't say much because of all the NDAs I've signed, but the companies responsible for chlorantraniliprole are quite interested in this. Now, fortunately, Reed Johnson, Ohio State, pictured here staring wistfully at an almond orchard, has already done a lot of work on chlorantinilprole in bees because this is the insecticide that almond growers use during the almond bloom. So most of our honeybees are already exposed to chlorantinilprole in large amounts when they visit the almonds. And Reed has done a lot of work on this because when it's co-applied with a fungicide, it becomes very deadly to bees, as shown here, where these tank mixtures can sometimes be an issue. But also means Reed's done a lot of the work prior to me becoming interested in this, looking at how chlorantinilprole gets from the pollen into, for instance, larvae. And the left-hand panel here shows the step down, the detoxification from pollen to nurse bees to royal jelly, about 100,000-fold reduction in concentration, moving from something like a protein patty to the larva. So if we're worried about larval toxicity, we have to remember that the dose received by the larvae is only about 100,000th the dose that we put in the protein patty because the bees are so good at detoxifying it. Reed's also done a bunch of standard toxicology, and the one I want to draw your attention to is the Altacore, which is the top left-hand panel here labeled A. And you can see there's no dose response curve there because no matter how much Altacore Reed fed to the bees or sprayed them with, you see no increase in mortality. You see a perfectly flat line, even at more than 10 times the field concentration. So somewhat accidentally, in trying to see whether this is a problem for bees, Reed has done a lot of the groundwork showing that it's probably safe when mixed into pollen patties, as shown here from one of our colonies a couple of years ago. I've got preliminary data from an experiment that just wrapped up this month that we started last year in April 2023, where we had 60 colonies across 4 apiaries. I believe you're recording this, so I've put all the information here in case anyone wants to go back. But basically, we just fed colonies protein patties with or without this chlorantinilprole, or no protein patty at all, from April through November, so most of the year. And we assessed them for varroa, beetles, brood area, you name it. And we're analyzing this currently. This was work done by my PhD student, Ian Collins, who's part funded by Project APISM. You can see here on the left just how bad our beetle infestations get, even by the midpoint in the year. Our traps were emptied weekly. So beetle traps were emptied weekly, and this shows you just how many accumulate inside a week. What we routinely saw was that the untreated pollen patties quickly became infested with large numbers of beetle larvae, whereas the treated ones remained larvae-free, but we could observe beetles laying eggs on them. So the beetles don't avoid the pollen patty. They, as far as we can tell, don't detect that there's a pesticide in there. They lay their eggs in, and then as soon as those eggs hatch and they begin feeding on the pollen at their second instar, they die. And we saw no single instance of our treated pollen patties being infested with larvae. Some early graphs here showing brood area, which kind of highlights that brood area is higher when you're feeding protein compared to not, perhaps unsurprisingly. That translates, interestingly, to quite a big difference in varroa numbers, because the larger brood area come fall means that the varroa aren't as quickly concentrated onto the adult bees. So late season, we actually see an indirect easing of varroa parasitism on our bees because the bees are able to maintain a larger brood area for longer. The patties tend to be eaten at a similar rate. What we don't know is how much of the difference between the treated and untreated is actually due to beetles. And then the final most important one here, the figure 4, shows that even just placing this treated patty, which is a very low dose, a dose we didn't think was capable of killing the adults, only killing the larvae, significantly reduces beetle numbers below the control. And when untreated protein patties are put in there, it exacerbates the beetle problem. What we're trying this year is mixing up formulations of small amounts of protein patty with a much higher chlorantraniliprole dose that we can place inside beetle traps like this one or this one to essentially act as a bait trap that the bees can't reach. And that's work that is ongoing as of this week. So I look forward to seeing if we can come up with an additional use of this chemical, not only to protect protein patties that we intend to feed to the bees, but also to create a much safer legal bait to place in traps. I will clarify that nothing I've talked about actually uses the Scott's Grubex that I bought from Walmart. That was just the very first time I tried anything. We work with technical grade pure pesticide, which is not legally available yet, which is why we're working on a formulation with the pesticide company that produces this. So cute as I find the adult small hive beetles, to circle back around, hopefully we're going to be seeing less of them in Georgia in about 5 years' time if this technology works as intended. And with that, I think I have about 10 minutes, 15 minutes to answer questions. So thank you all for your attention. I'll answer the ones that are in chat, and then anyone else can pitch in as they see fit. So someone else, is powdered pollen as bad as patties? We tend to find that the bees consume actual pollen faster, and that because it's a little drier, the beetles don't get into it as easily. If you put a large amount of pollen in there, they will still breed in it very comfortably. The problem with actual powdered pollen is it's so expensive that for a lot of my commercial beekeepers, they can't be throwing that in there, really. So one question is the quality of the container important to reduce egg laying areas? I think by container, we mean the hive body here. Would that be a fair assumption? In which case, the beetles they're always going to find some hidey hole to lay eggs in, whether that's the edge of the frame, in the tiny gap where maybe the foundation wasn't properly sprayed with wax. They're always going to find a crevice, just about. Really, it's whether that crevice is anywhere near things that the beetle larvae can eat. That's the deciding factor. Otherwise, happy to take any further questions. So someone's asked about combat. Is combat fipronil? Is that, I tend only to work with the scientific names. But if combat is the roach brait, which is fipronil, it is wildly illegal to put that in your beehives, partly because it's very dangerous to the bees if they're exposed to it, but also it's really dangerous to people, whereas the chlorantraniliprole tends to have really high field use safety ratings. It's used very abundantly on soybeans at the moment because it tends to have a very promising ecological and kind of health impact.
Daniel, back to that last question. Was that what you were indicating by quality of the container, that the overall hive? Was that what you were referring to?
Yeah, we find that and ground control to be the best way to take large infestations, out of an apiary.
Okay, thank you for clarifying. I'm sorry, Lewis.
No, of course.
Was there another question that's come in?
We've got a couple more. So we have, are there generally accepted methods to monitor beetles, similar like the methods we have for Varroa? The only one we reliably use are the kind of beetle blaster traps with the oil that we can use as a lethal sampling. Right now, we don't have great ways to do something like a Varroa wash. There's been a lot of work on seeing if there's kind of pheromone traps that can be placed, but not with great success. So we don't have as easy monitoring of them as we do with Varroa right now, which does make doing the science kind of difficult sometimes, I'm not going to lie, but also can make it hard for beekeepers to assess whether they've got a problem.
They're very light adversive, so as soon as you open a colony,
precisely. They scatter
prison
so
break. They're out.
quickly that short of taking a photograph as soon as you pop that top lid or that inner cover, you're really not getting a good metric. And even then, so many of them hide inside the empty comb, like drawn up comb, that I wouldn't trust even that. I really think trapping them is the only meaningful way. You could probably get a reasonable estimate off sticky screens, but only if you've got really heavy infestations where you're getting a lot of beetles dying naturally inside the colony. In cooler areas, can the small hive beetle mate late fall or over winter if infestations are found in the fall? I don't know that they can mate once it gets cold, but they do overwinter reasonably well. I will say that on 2 years ago, well, a year and a half ago, we had some really extended cold weather here in Georgia that caused a proper ground freeze for a few days, and we saw far fewer beetles for the first couple of months of that spring. So wherever they're overwintering, they're definitely vulnerable to a kind of a deep freeze. We didn't seem to get one of those this year, so they're kind of quite abundant even this early in the spring. And in those more tropical, subtropical regions, they almost never get actual ground freezing. But in regions where that does happen, we see it knocks the overwintering population down very heavily. I have a sneaky suspicion that the majority of small hive beetle overwintering actually happens at that pupal stage, and that they're not doing it as adult beetles, apart from occasionally inside colonies overwintering alongside the bees. Hey, Mad Hat, yeah, I'll ask you in a minute. Good to see you. I have a feeling that the majority of overwintering is actually happening pupal in the soil, and then it's a new generation that emerge in the spring and get to it. I don't have anything to back that up. We just know that's how a lot of other insects work.
Medhat did want to also make some comments on that, I think. Medhat, you're muted.
Yeah. Oops. Thank you for Luis. That's excellent presentation. In terms of small hive beetle in northern climate, actually we found them. They survive. The adults survive with the cluster of the bees, either indoor or outdoors. I think the cluster warm enough to keep the adult beetles happy and survive the next spring.
Yeah. I think in regions like yours, especially where you get huge amounts of snow and very cold weather, I think the only place they can survive is in with the bees,
Right.
for certain. We have Manuka honey has high concentrations of hydrogen peroxide. Do New Zealand hives have few or no beetles? I don't believe small hive beetles exist in New Zealand. I don't think they've arrived there, so we haven't been able to test that yet. But I don't think small hive beetles have ever been detected in New Zealand. I think they're still SHB free, so I don't think we can answer that. Do SHB have droppings that can be seen on inspection trays? A method of monitoring? Not that I know of. I think the larvae probably produce some meaningful frass, but if you're seeing that, it probably means the infestation's already kind of in dire straits. The adults, I don't think that I've ever seen produce meaningful leavings that we could detect. Not that, and we've raised them in large numbers in the lab, so I feel like we would have seen it. It's not like bee droppings or anything that's quite obvious what it is. Nelda asked, I may have missed this. Do you expect the pesticide and trap to be suitable for use with honey supers on the hive? That is a question for the regulators and not for me. I think my expectation is they won't want the chlorantinilprole in there when honey supers are on the hive, but there are already really well-established tolerance limits for this chemical in food because it's so widely used in row crops. So I might be pleasantly surprised, but I am not familiar enough with the inner workings of the EPA to be able to confidently answer that yet. I would like the trap formulation to potentially be able to be used year round. I think the protein patty feeding, I think it would be understandable if they said only when honey supers aren't on the colony for that, but that would be my speculation. And yeah, as Daniel confirmed, they're not present in New Zealand. They're absolutely terrifying in Queensland, in Australia, on the Gold Coast, which has a very tropical to subtropical climate. I know they have horrendous problems with them there. Yeah, we've got another couple of minutes if any questions abound. Otherwise, I will happily leave it to the next speaker. My
Are
email's
there any
also
other
right there
questions?
if anyone wants
Thank
to
you.
reach out with anything later.
Yeah, appreciate that. Thank you. Are there any other questions? Anyone care to unmute and ask questions? I think we've exhausted the questions.
All right, well,
Heather, actually, another question: is mating in the air or in the hive, or?
it is mostly in the hive, we expect, given small hive beetles really don't like flying, honestly. They won't if they can avoid it, which is why when you open up a colony, they tend to scatter rather than just fly off. From what we've observed in the lab, accidentally, not intentionally, they mate very freely in the kind of warm dark of a suitable place to lay eggs. I think there's lots of cool work to be done on them from a biology perspective of how they kind of compete for mates and all that kind of stuff. But that's more just me being a nerdy bug person than anything that's useful to beekeepers.
Is there an alternative host for them? We've often talked about this being an area where, you have crops on the ground, et cetera, watermelon, melons, et cetera.
Yeah, so there's speculation that they like fruit and such. And I've seen that someone's just asked that. I have
Oh,
never
yeah.
seen any evidence for that personally. I think maybe very occasionally the smell of rotting fruit and that fermenting yeast perhaps gives off something similar to a hive. I'm much more concerned when it comes to secondary hosts about the frequency at which they're infesting wild bumblebee colonies. And that's work that one of my other PhD students here has just started. We actually just got bumblebees delivered to the lab today for the first time in many, many years. And we're going to be seeing if small hive beetles are capable of invading those. Because I think that's not only important for understanding where else in the environment they persist, but also I think it increases the importance of us controlling these in our honeybees if they're also a threat to things like bumblebees. There's some evidence that they do, but nothing definitive. So that's something that we're working on this year.
Okay. Before you go, a quick update on the yellow-legged hornet. What's happening in Georgia with that nasty?
Lots. So we have thousands of traps that are being deployed. We have myself, we have USDA APHIS, we have the Department of Ag, all throwing people into this. None of us are really expecting to see any crop up, honestly, until May, June at the earliest. The later in the year, the larger the colonies get, these things grow exponentially up until about November, October. And right now, you're really trying to find a needle in a haystack. But we found 5 last year, all within a very, very small geographic area. We have evidence that we missed maybe 1 or 2. But we've now got a lot more practice and a lot more forewarning. So my teaching finishes at the end of April, and the rest of my year is going to be spent trying to get these things off the Americas, because I already have enough research I want to do without having to become a de facto yellow-legged hornet researcher here in the US. These things actually invaded Britain right before I left for California, and I was in the very far south of England working with Pete Kennedy, who championed the use of the radio tracking for these things. And I was in the same building as him when he was first working on these back in Britain around 2015. And now they've followed me, and I am very determined to get one up on my European colleagues by getting rid of them here, which they've failed to do over there, but we've caught them extremely early. So I honestly do think we have a good chance of pushing them back into extinction here in the Americas. But more than that right now, I can't say. We're kind of in the off season until they start cropping up.
Yeah, thank you. Thank you for that update. And thank you too for the great information on the small hive beetle. Appreciate it
Gladly.
very much, and the work that you're doing in the lab there in Georgia. Appreciate your joining our WASP mini conference this evening.
No,
Thank
of
you.
course. Thank you for having me. Thank you for the enthusiastic questions, and enjoy the rest of y'all's conference. Thanks, Stuart.
Okay, thank you. We're going to take a quick break so we can get Ethel's slide deck up and share screen. So about a 3 to 5 minute break here, so that you have a potty break if you need it. Get another beverage, and we'll soon have Ethel on. Okay, you were able to get your slides up.
Yeah, just trying to get it to be in. Where is it doing that? Okay, hang on. Give me 1 sec. Okay, no. I want to have it. Hang on a second.
Let's go back. Yeah.
Yeah. Stop share. Let's see. Have it in full screen. No, I can't have it in full screen. Okay, hang on.
Bring up the way you had it, and then go to slideshow. That would be one way to do it.
Yeah. So
Start
it's
from
just
beginning.
unfortunately, yeah. Hang on. Let me, if I just put it here, it will be fine. And share, and voila. And why is it so teeny? Okay, I don't want this part. Okay, hang on. What is going on today? Hang on. Close that. Close. Okay, slideshow. Play from start. Okay, we got it.
Yeah, now you're full
Yep.
screen. Yes,
Yeah.
excellent. Okay, we'll give another couple minutes for people to get another adult beverage or beverage of choice and potty breaks. And what branch are you with at the university?
The college I'm in is the College of Tropical Agriculture and Human Resources. So there is a entomology department as well as a plant disease department, and so we're kind of split into a couple groups. And there's also people who work more on soil managements and things like that. So
Okay, is that on Oahu?
it's on Oahu, yes.
Yeah.
And I think, I don't know if Micaela is still there. The second person that, yeah, there she is with her name. So I don't know if you want to say hi to Micaela. Micaela, you there? I guess she might be muted. She's the co-author for some of the work we did on the actually she's the lead author for the work we did with the little microplastic, the Swifter sheets or shop sheets as people call them. But I also, as I was listening to Lewis, I decided to add a few things because and I would love to talk to him at some point. So we definitely see beetles throughout the year since we have a very subtropical climate, and we've been working a lot in terms of the densities and the peaks and no peaks and management. So I don't want to make my talk about that, but I did add a few notes so that we can have a forum to compare notes. So it'll be fun.
Yeah, exactly. Yeah. Lewis is very, very approachable, and I think you'll find you have a lot to try to share of
No, no, no. I
information.
think it would be great. It's just, I mean, it's obvious that Hawaii, in some ways, is its own little entity, because we're one of the few places that has European stock in subtropical climates. Everywhere else, you end up with a lot of Africanization running. So, we good and bad. Some of our things are a little bit more relatable in the sense that our bees are pure European stock, but it would be interesting to see, get some dynamic. I mean, people have their own opinions, and it would be great to get some of the participants also to share their experiences or ask their questions. So that'd be great.
Okay, I think we'll get going with our second of our presenters this evening. That is Dr. Ethel Villalobos, who is at the University of Hawaii in Oahu. She's here with a co-presenter this evening as well, or at least on the publication of Dr. Migala Budler from Argentina, I believe, right? And,
Yes.
yes. Dr. Villalobos has worked in a number of different countries, and is also very active in terms of not only bees but also the social wasps as well. Published a paper that I found fascinating, and that is small hive beetles as a source for microplastics. As a past president of WAS, it's an honor to welcome you back to our WAS and to our mini conferences for your presentation tonight on management practices, small hive beetles, the source of microplastics. The floor is yours.
Thank you. Thank you, Joey. That was a very nice introduction. Thank you. And I will try to keep track, but Dewey, let me know if there's a question that seems super relevant and I'm like focused on something else, because I think integrated questions sometimes work best, and I can always speed up my presentation at the end. Okay, so I would like to start, let's see, start with, so Lewis already covered a bunch of things about the beetle. We know that it's an invasive species. We know it has moved across the world, all over the place, and it keeps popping up in different places. And part of the reason that, say, Argentina is now concerned is that it's popping up in South America, very likely multiple points of introduction. And South America is such a diverse continent that you end up with, climates that are very tropical, climates that are very dry, climates that have some mountainous places. So the biology and what to expect from such an adaptable little beast is kind of tricky. So we're hoping to produce data and information that is suitable for a wide range, but everybody's going to, and that applies for the US beekeepers as well. What works in Florida may not work in Georgia or in Texas or, further up if you go into the Carolinas. So I think it's great to have a foundation, but trust your eyes, trust your observations, check and see what you have. And the small hive beetle is actually a interesting invasive species. It does live inside the hives. They do produce aggregation pheromones when they're ready to reproduce in mass, which doesn't happen very often. And this is one thing that I want to point out. So this picture here gives the impression that you have a couple of beetles, and then all of a sudden you have this huge mass of basically slimy worms, and the whole colony is lost. But here on Oahu, we have colonies that have had beetles for years and never had a slime out, never had a loss. So reproduction does take place. Biology says they do what is called cryptic reproduction. That is, a female will scurry around, go to a cell where there might be a developing pupa, can put the ovipositor, puncture the cell at the bottom, and lay some eggs. So very rarely you would open a cell and instead of a bee larva, there would be beetle larva. But this is super rare. But the beetles also live very long time. So you can replenish your adult population with this trick. So you can have continual cryptic reproduction with sudden peaks of mass reproduction. When the beetles are going to have mass reproduction is when these aggregation pheromones come in, because then you call everybody, you basically have a giant beetle orgy for a few days, and you coordinate egg laying. And the success in mass reproduction is you overwhelm the bees. You have so many bees, so many larvae, so many eggs, that basically the chances that your egg or your larva will get male is very small. So it's a great strategy in a sense, but it happens infrequently. So when it happens, it can be huge. And when a colony goes through this, it also delivers to the environment of a very, very large number of beetles, which is very problematic. So one thing that I do want to focus on, again, kind of leading to the use of traps, is that the beetle distribution, remember that they came from South Africa, they lived in association with feral bees that were, say, inside a tree. And when you have a feral colony, you know that they will have basically the important stuff. The brood and the pollen will be in the center. And as they grow, they add combs for honey. To be honest, the same is true in a managed colony. You find a brood towards the center, and as they grow, they fill in the external frames with honey. Now, the beetles, what the beetles would love to get access to is the high quality food, that is the protein, because that's important for reproduction. So brood and pollen, it would be the ideal. But brood and pollen is where most of the bees are. So the protection in the area of brood is much higher. So often you find the beetles relegated to areas on the periphery. That is, you'll find them in honey frames, you'll find them scurrying around sometimes on the bottom board, sometimes, and we'll go into that in a little bit. But in general, the basically the goals of the small hive beetle is to obtain something that will allow them to have very effective reproduction. The bees, on the other hand, get in their way. A bee cannot kill a small hive beetle. They chase him, they pursue him, but they really suffer from short attention span. They will chase him for like 10 seconds and then forget about it and go on doing something else. So they're not really threatened inside the hive. They're really not. So it's disruption more than anything. And some of our traps that we use rely on that disruption and that behavioral patrolling of bees that will drive the beetles to certain areas. And as Lewis was saying, the density can really vary. You can have a few beetles, you can open a colony like the one on the right, and you can just have a complete scurrying around of beetles all over the top, the sides, it's scurrying down away from the light, but you can have massive numbers of beetles. Currently, and this is we're at a low point compared to when the beetle first arrived to Hawaii. On average, we probably, in beetle blaster traps, we probably capture at least, I would say, around 20 beetles per week. So 80 beetles, okay, let's say 50 beetles per colony on average on beetle traps. There's still, if you were to examine the colony, there's still a fair number of beetles that haven't quite fall into the trap. So the numbers per week are quite high. So we have high densities. There were points when the beetle first arrived that we were collecting 100 beetles per trap per week. And over time, the numbers have decreased. The colonies have become better at managing the beetle, but also the beekeepers have become better at managing the beetle. So one thing that we discovered is if you go back again and try to look at the biology of the beetle, in a feral environment, a colony in a tree trunk will have combs hanging from the top. Debris would fall at the bottom, which makes the bottom, the debris filled bottom, a pretty good place for beetle larvae to reproduce, and really not a place where the bees hang out. They may try to clean and empty out a little bit of the bottom, but it's not as intensely clean as what you do when you have a bottom board in a managed hive, like on the picture on the right. So the different thing that we encounter is that in a hive, in a feral, in a cavity, whether it's a tree trunk, a cavity in rocks, the top is the top. There's the ceiling, there's your comb, that's the top. Us as beekeepers, we can add layers. We can add a second story, a third story via the honey supers. This gives a very different structure, because in a honeybee colony, in a well, in a feral, in a natural comb, the bees will tend to protect more at the center where the brood is, and the beetles will end up being pushed to the sides or to the bottom. In a managed bee colony, what we found is that the beetles get pushed more to the top and to the sides, usually to the sides of the honey super. The bottom, not as much. You find, and we're preparing a publication, we have one in review right now, that basically points out that the accumulation of beetles at the bottom happens during your manipulation, during your colony manipulation, because you make them react to the light, you make them react to the smoke, they go down, and then you find them at the bottom. But if you were to lift a whole colony before you do anything and you check the bottom, there will be very few beetles at the bottom. So again, all this kind of biology, where are the resources, where are the beetles, what do the bees do? All this is very important for management, because it gives you an idea of where do you need to sample and what can you use. And one of the things that I have a little video to show you, you open a colony, and again, Hawaii has colonies that produce lots of propolis. Some of these were beetle jails where the beetles were trapped inside. And when you take out the inner cover, everybody scatters, but there's some of the beetles, going and running around, and that's just a few of the beetles. So this is what the bees are encountering every day. They're having to deal with these guys. There's no winter, there's no break, it's just beetles. Does it rain? Yes, we have a pretty wet area. Soils in Hawaii allow for pupation, but some soils where people thought were not going to be good, like lava rocks, we actually tested beetles in jars with lava rocks, and they would find a crevice in the lava rock and spin a cocoon and pupate in there. So the ability of this beetle to survive in different conditions is huge. It also, I don't have in this talk how fast they go underground. I have filmed larvae in containers, and in less than 10 seconds, they're gone. They basically screw themselves into the ground and they're completely gone from the surface. And they can go pretty deep. They can go 20 centimeters down. So that is a problem when it comes to management via chemicals in the soil, because if you have rain, it's going to wash it off. And some of these chemicals are not great for aquatic organisms. So streams, lakes, things like that. So you may end up, kind of recycling it back into your colonies or affecting other organisms. And also maybe a waste of money if you are applying chemicals that get washed out of the soil when these things move so quickly and so deeply. So one of the things that we've been trying to do is, and this was done in Hawaii, in Costa Rica, and in Mexico, with very different densities of small hive beetles. And what we were trying to find is something that was asked in the previous talk was, is there a quick way to sample for beetles or is there a beetle threshold? Okay, no one has been able to establish a beetle threshold. We had colonies that had 500 beetles and did okay until something happened, until people harvested or until it let go of a swarm, and then the space to bee density changed, then things happen. But it's very, very variable. So what 30 beetles can do to a small hive or 50 or 100, it's very variable. So what works, I think, is to have an idea of the relative density in your apiary. So if you can establish a baseline and you say, okay, well, now I found this many, and 2 weeks or a month later you find 3 times that number, then you start worrying, then you start figuring out what you can do. And what we wanted to prove was, is there a shortcut? So what we did is we developed a technique to just very quickly with various observers just to establish how many beetles we could catch. So we would take out the cover and inner cover if there was an inner cover, and we would capture and kill all the ones there, and very quickly also take the side frames. Those numbers, okay, which we call the tops. So there's in the graphs there, you have the top to Hawaii, the top in Costa Rica, the top in Mexico, and the top in Mexico minus one outlier, and I explain that in a second. Basically, there's a positive correlation. What we did is after we did that, we went through the hive and tried to capture and kill most of the beetles that we could find, and we determined where they were. Were they in the brood frame? Were on the sides? Where they had moved already to the bottom? And basically, with the numbers that you catch on the cover and on the side frames, you can guess how many there are. So it's a pretty good relationship. You can more or less know if you have a high infestation or not. And this works really well if you, and this worked for the Mexico one. One thing that you have to realize, the colony that was the problem here had 300 beetles that we captured in that colony, and it was a single deep. That colony was completely overrun by beetles. So they were running all over the place. So our capture on the top and the sides, I'm sure, went to pieces because there were so many beetles, but that was an extreme. If you take away that colony, the relationship is much stronger, similar to the other ones, meaning the more you have on the top, the more you have overall. Is pretty reliable, and you can use it as a relative number to estimate how you're doing in your apiary. And one thing that you can do that adds to the efficacy, and it works really well, is if you get a piece of felt and you brush it with like a wire brush and make it just slightly little fussier. And if you're sampling your side frames, those will have mostly honey. Unlikely they will have a queen. Unlikely that you're going to have too many bees. You can do what I'm about to show you. And then there are beetles there. And like Lewis said, they usually walk. They try, in this case, to run to the bees. And I can play this again. And the cool thing about this method is that you see that one crossing all the way, running to the bee group. A bee tried to stop him and then decided, whatever. And this is actually a really good method because you can easily squash and count the beetles. The bees, at the end, you can just shake the felt and they'll fly. Very few bees, like 1 or 2 maybe, they'll get entangled. And it works really well. Hang on, now when I go to the next slide, this is a slow motion version of that. I don't know why the slow motion changed the color, but there it is. So there's the beetles walking, and basically you can do this very easily. So if you check the top and you have the felt there, you can put it on the cover, and then you can bang your side frames, and you will have a relative measure that is very accurate of the total beetles in your colony. So this is as simple as it comes, and it can work. So that's one thing. Now, the importance of relative densities. Why are we concerned? Because it gives us time to act. Again, if you had 10 beetles with this method and then you went back and you had 60, then you know something's changed. Either your colony is a little weaker, it has too much space, what's going on? So you can then act, and you can see the picture on the back, on the right-hand side of the hand, and covering beetles. And this is what we face here in Hawaii. So, but again, the beekeepers have gotten better, and the bees have also gotten better dealing with this little pest. Now, the management strategies can be divided if you're going for tools to use against the beetle. They're divided in a couple different kinds of things. One is the traps on the top, often filled with oil. They sit between frames. Not all of them are used with oil. Some people put, I've seen people put diatomaceous earth. I've seen people put chemicals and with pesticide in it, all sorts of things. We just use oil. You only need like a tiny little amount of oil because they get tricky when you move them and you try to get them out. The other method involves the floor of the hives. And what we find, and okay, every single trap that is in this picture will catch beetles. The problem is, how many versus how many there are in the hive, that's the debatable part. Based on what we find is, and people in Hawaii, when the beetle first got here, went crazy over screen bottom boards and large pans of oil, which was incredibly difficult because the bees will come and propolize things. And unless your colonies were in a perfectly flat, like parking lot like area, if you're trying to move this and there's a little propolis, a little wax, you're going to splash the whole bottom of your frames and the hive. So it was, most people moved away from that, but it was a really common, very, very popular means. Then it became those, modified CD cases or the traps, the beetle barn traps, like the one there in the picture, the one that Lewis showed, the white one, which, there are many versions of that. And a lot of people were using fipronil as combat, as roach food. They would put like a little bit of pollen patty with the combat in there and you would get beetles. I wasn't planning on discussing pros and cons of things. I'm just telling you a couple things. Some of these chemicals go through the plastic and you will find basically marks of where they were that actually are contaminated wood. So if you put in something like fipronil, be really, really, really basically move away from that. They also, all these traps, the beetle traps, the beetle blasters, all the traps with oil, including these traps here, the bees seal with propolis. So just because you slide one of these things in, don't think your work is done. There's a lot to be done with management. And to be honest, most beekeepers in Hawaii have moved away from traps. Most of the beekeepers are managing the bees and beetles, even with these high numbers, based on space and density of the bees. This, of course, is taken a while and the climate is different, but just saying, we don't tend to feed the massive amount. The picture of the pollen patty that Lewis showed, there was a big pollen patty that he flipped with a lot of beetles. We would discourage beekeepers to feed chunky. We tell them to feed flat, to squash it, because if you allow for the bees to have enough bee space where they can actually walk on top and where it's very narrow, they'll find the larva and remove it before they get huge. And this happened to a student of mine that was doing a study on foraging, on feeding bees, on supplemental feeding. The moment you put fat pollen patties, the beetles were smart enough to put the larvae were sandwiched between two pieces of food that protected them. The moment you narrow it down, the amount of use of the pollen patty was greatly reduced. So thin pollen patties work better. In our case, we don't have huge colonies, so 100, I think it was 100 grams per colony per week, so like basically almost like a third of a pollen patty would work. If you flatten it and you give it on a weekly basis, that's what it was working for us. So among the treatments that people have started to use was the microfiber towels or sheets. So they look something like that. And they definitely were, when they started, and now Man Lake sells a version of them, they were basically brownie shop towels. There were people insisted, the beekeeper insisted, had to be the blue ones. And the idea, the funny thing is, when I look back, I found that in Bee Culture in 2015, there was a review on the use of these towels, and they were saying, oh, I love the simplicity, it's simple, non-toxic, and cheap. And the problem with this is, yes, sure, it's simple. And like all of these traps will catch beetles. So the impression that we see a beetle in a trap gives us this kind of peace of mind that is working, but we don't know how many there are there. We don't know what the situation is. So in a sense, it may give us a false impression of what's really happening. Then the non-toxic is like, if we don't see immediate death, we assume that it's okay. And I was always concerned about this and never had the know-how to test what to do until Micaela came to Hawaii, and she is an expert in pesticides and chemicals and bees. So we decided to, and she's done work with microplastics in Argentina and bees. So we decided to look at what is happening with these little towels. So before we start, and this is in the apiary area in Hawaii and Oahu, and was to look at what we call microplastics. And basically microplastics are just things that are small, so smaller than 5 millimeters. If they get smaller than 2, they call them nanoplastics. So tiny, 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 tiny pieces of plastics that, can come even from the fabrics in our clothing. I love my fleece, but now I feel guilty every time I use one. But they are everywhere. And part of the reason Michaela wanted to come here is because the ocean is a place where people have done a lot of studies with microplastics. So aquatic systems are filled with microplastics, but the soils, the way we farm, we use a lot of microplastic materials for planting, for irrigation, for things like that. And they're in the area, can be found anywhere, including the Antarctic, Arctic, they're everywhere. So when it comes to that, then you go, well, are we talking about one more tool? What is the importance? And what we decided was to really take an in-depth look at this. So for some of you, and Lewis mentioned that the bees are really good propellizers, so are ours in Hawaii, depending on the colony and where it is. But some of them now propellize the towels really fast, some are not so good. And you will see something like this, like the picture on the right with the strip and all the little dead beetles. And then the tendency is like, oh, you see, it works. And it's like, okay, how many beetles are in the hive? What's happening here? Of course, it's catching some. So, but what's going on? And then what we found, and in this video, I'll play it twice, so I have a chance to say, you can see this beekeeper keeps a screen, don't ask me why, it's kind of not a queen excluder, don't ask me. But anyway, he keeps a screen. And the little towels were underneath the screen, and down there is the deep. But the interesting thing here is you can see the bee is trying to chew the fibers and remove them. The beetle's spinning around because one of his legs is caught. So they do get stuck. I mean, there's no doubt, but look at that bee coming, inspecting him, yeah, whatever, moving on, so they are not really individually targeted by bees, they're chased. And hang on, I need to get out of here to move to the next, no, I want to move to the next slide, hang on. Okay, hang on, let me escape for a second. And go to the next. Okay, so this is another video, and in this one I want to show you something else. In this one you can see in the back a couple dead bees, and the bees will come and remove dead bees. So when you see a towel that is used and you see it's much easier for them to remove a dead bee than it is to remove a dead beetle. So you also underestimated the number of bees that get caught in this. And so I see the one in the back is trying to remove dead bees. They'll come and drag them away from this area. They want to get rid of the little bodies. So why do we worry so much about this method? Well, one of the problems is that we really don't know what we're using. If you buy these things, there's no like list of active ingredients like there would be in a pesticide. It's just a sheet that you put. So the shop towels, the original brownie blue towels, were definitely used off-label. They weren't obviously designed to be used in beehives. The Swifter sheets that people have mentioned have been widely suggested in the beekeeping forums. And then now there's some by Beekeeping Supplies that really don't say what they are, okay? So very, very, very little info. And sorry, there's a little bit of Spanish because I'm supposed to talking to a Spanish group. So what are these towels? Okay, well, it's interesting because they are created through a process called hydro entanglement or hydroknit. So they're basically a bunch of plastic fibers. They get pressure washed into a sheet. So they look woven, but they're not. They're just entangled. So they're like a super, super strong, messy mass of fibers, okay? And they're advertised as resisting tears, cleans the grease and grime. It's okay. So resisting tears and cleans grease and grime should give you 2 points of concern. Resisting tears is not like when you put a newspaper to separate 2 colonies and you're hoping they'll eventually join and they'll tear the newspaper. These things are hard to tear. These things are hard to rip. And I'll show you what the bees do. Now, cleans grease and grime. All tiny microplastics tend to, in a sense, retain a lot of things. And now there's a great concern that some microplastic serves as a substrate for things like bacteria for growth. So they do provide that kind of surface. And what you see when you put a towel is over time you're seeing this. So you see and you go, oh, well, look, lots of beetles and they're cleaning it out. But again, naively, I think we all think they're cleaning it out and they're throwing it out of the hive. But you really don't find like a pile of blue fibers outside, and we don't know why. But something is happening. But, well, we believe the bees are somewhat ingesting the dye, but the color does decrease over time of these towels. But you don't see a pile of like little, fibers outside the entrance or near the hive. So what we did is we looked at the fibers. This is on the left is a brand new towel under the electron microscope. And you can see that they're not woven, they're just entangled. And this is the way they're made. And then on the right, you see one that is being chewed by bees. And what you can see is that the fibers, they can't rip them. They can't cut them like a scissors would on a thread. They're peeling, they're shredding this way, the long way. That means they're making them into smaller, long pieces that, guess what? They're consuming. So what you're going to see in this video is a bee actually trying to remove a piece, the one underneath the wire mesh, well, several underneath the wire mesh. You can see that they're pulling and pulling and pulling at those fibers. And these bees are going to be ingesting actually those fibers. So they're not just like carrying, like a dog will carry a bone and throw it outside. They are actually eating it. So what we decided to do was to do an experiment, and we borrowed some hives from a producer that uses the shop towels. We oh, sorry, this should be control near. So sorry, go back. So this should be control near. Is our apiary, the UH apiary, and control far, which is only 1K, so less than half a mile away, is a producer nearby. And so we wanted to see if we could track the fibers from this beekeeper to ours, which is very close to another beekeeper's apiary that is farther away, and see if we could find these fibers. And this is the result. So what we did is we caught bees. We caught nurse bees in the hives, in the treatment colonies, the yellow ones, which had a higher number of microfibers. And what Michaela did is we looked at the surface. So the wings, the legs, the fuzz, what was in the surface of those bees that we caught, and the nurse bees that we got inside the hive. Then we look at the control near, and we found the number of microfibers were less. A lot of them were did not, and the control far even less. So these two, however, the control near and the treatment were not different. So that means that what the beekeeper is doing with the towels is affecting my bees. There's enough drift that some of the bees are going back and forth, and my colonies are having the same situation as him statistically. The 1 kilometer away, say half a mile away, those colonies did not have an effect. So their bees were cleaner. We also looked in honey samples. So we looked in the, oh, sorry, go back here before Micaela kills me. And this one, we also looked internally. So she also did digestive system. So we know they also had microfibers in their gut. So outside and inside, the bees in the treatment are contaminated, let's say, with the microfibers. When we looked at the honey, we collected honey in the treatment colonies, in the control near, and then control far. And in this case, the honey and the control near had a much higher fiber concentration than my apiary, say the UH apiary, and the collaborator apiary that doesn't use the fibers and is half a mile away. So that was at least encouraging, but obviously there's an effect. The fibers don't disappear. One thing that we didn't do, but other people have looked into, is what happened. Where did the rest go? How much of that is getting into the bee larva, into the food for bee larva? One of the things that, and this, I cringe because Micaela knows this so much better than I do, but I'll give it my best shot. So what we did is we collected fibers that were on the bees, but we also had fibers from brand new towels, the same kind that were being used. And these are, okay, every single plastic reflects UV light. So using Raman analysis of the microfibers, we can create graphs, peaks that tells us what this is. So they're giant, basically books that says if it has this pattern is this type of fabric, if it has this pattern is this type of plastic. So one thing that we found is this is from the brand new fibers. Okay, so PET is the dye that gives it the blue color to the fibers, and there's the dye. Okay, and this is oh no, sorry, this is the dye PB. Micaela, you're making me nervous just being there. Micaela, you want to do this? So okay, sorry, my bad here. So these, sorry, this is the dye. The sorry, the PB15A is the dye, and the dye was present, and PET is the polyethylene fibers. So this is the plastic, like basically polyester, that those things are made of, the little towels. And you can see our samples reflect the plastic, the constitution of the plastic. So our fibers collected from the colony, the fibers collected from the inside of the guts of the bees, the fibers collected from the honey, they all match the structure of the fibers from the new towel. So we have a pretty good idea. There were additional types of fibers in the colony, as is to be expected. One of the things that is important to know is that the impact of microplastics is now really, really being studied. It started, in a sense, a lot with aquatic organisms because the plastic is floating in there. And we know that it affects vertebrates, fishes, for example. And it can affect the feeding behavior because basically they will be ingesting all these things and they lodge in their stomachs and their intestine. The dyes that are used in these fibers are also being ingested, and we don't know exactly what they do. The other thing that is interesting is people have worked mostly with other insects that are maybe easier to keep in the lab. And we know, for example, if you feed crickets, which have been a wonderful thing for study for many years, for many entomological pieces of work, if you feed in microbeads, which are really tiny little round pieces of plastics, it didn't affect them as much as if you feed them fibers. And fibers tended to wrap around the gut and wrap around the pupa and reduce the growth rate of the cricket in general. So we know that fibers can be really detrimental to the growth. So we don't know what they're doing to the bee larva and how much of that is getting into the bee larva. We also know that like we found that polyester is common in bees, that you'll find it in wings and legs and hairs. This is a study, Ito et al. In 2020. Also, Michaela and colleagues worked on tracing the microfibers that this was an experiment, so they added microfibers to basically food for the bees, and they were then trace it back into brand new, for example, wax cappings, into the larva, into the honey, into the bee food. So we know that is being transferred and spread across the hive. The problem with this is it's very difficult to know, although there might not be acute toxicity. It's very difficult to know what exactly is happening. We know that microplastics can cross basically even in humans, the blood, say, brain barrier, it can go across. So we know that in bees it can accumulate even in their brains. We know it makes them more susceptible to viral infection. So things that we're trying to do is to provide good food for them, because that improves their immune system. But if we selectively using things that are maybe unnecessary, that basically is a source of microplastic in the hives, we may be undoing some of the good we do. And in the case of the small hive beetle, there's really probably no need to use these sheets in most places. So here, there's a little picture. You can see a little blue fiber stuck in the little feet of the bee. So we found that there's an increase in the number of fibers in the body of the bees. We don't think there's, acute toxicity. However, there's probably chronic sublethal effects that are very, very hard to detect. And basically, what we're choosing to do for the control of the beetle does matter. We can't control the environment. Microplastics are everywhere. But I think this also goes to taking a stand and, how we feel about plastics. And when Michaela was here, we had a meeting with researchers at UH that work on very different things. And some of them were even working on placenta. They were studying placenta, human placentas, and they were finding microplastics in placenta because we humans have bits of microplastic floating in our blood, and it's getting lodged. I just saw a very recent article where people that are having surgery for plaque and problems with their hearts and arteries that are clogged have microplastics in those blockages. So we really need to, I think the bees are alerting us, and it's a great system to study this. And it's a bigger problem than the microsheets for beetle control, but it does makes you wonder what you want to use in the colonies, and are there other ways to handle this. And I just wanted to say thanks to the beekeeper collaborators, to Pat and Tina in Hawaii, and this work was supported by Eva Crane Trust and Hawaii Invasive Species Council. And then I'm open for questions.
Fascinating story. Fascinating how it an observation turned into even many more fascinating questions than trying to answer the initial question that you had. Appreciate your joining us this evening. We had a couple of questions. In California, Daniel said that they don't tend to use the mineral oil because it goes rancid, but tend to try the diatomaceous earth more frequently. Do you have an opinion on in the traps, the beetle traps, one over the other?
Not personally. I know people who use diatomaceous earth. We have used the cooking oil and didn't have much of a problem. And to be honest, the beetles don't go to it for their love of the oil. they go by the chasing. So I think the biggest problem is the effectiveness of any oil trap will decrease if your super where you have them is less populated, because the engine that drives that is the antagonistic chasing behavior of the bees. So many times I find people going, no, I'm not catching anything. And it's like, you have no bees. The beetles don't need to go that far up, they can hang out in the second level. So I'm sure at some point our stuff went rancid, but I think it's more like a physical, it's gooey, it drowns them, it will go. People in Central America trying to save money tried soapy water, but that evaporated really fast. So that didn't quite work for them. But diatomaceous earth has worked for some people. I use the oil, never had any problems, but I also have to, we have to replace it, empty and replace. And we're very cheap. We reuse the traps as much as we can. We can get the beetles out and refill them because they're expensive. And but yeah, no, we never had any problem with rancid oil.
Okay. There on Oahu, you're talking about what you're doing your work there. Is a small hive beetle distributed on the other islands as well?
Yes, everywhere. It spread like wildfire. The varroa mite is only on 2 islands, thank God, in Oahu and Big Island. The small hive beetle is everywhere. And it was like in less than a year, it was absolutely everywhere. Because it can travel, it's sturdy, it can travel in equipment, it can crawl into your bee suit if people were moving things like, going to another island with equipment, it was really fast how quickly it went. And it was really horrible for the first few years, like horrible. We, especially in the young colonies, we had beekeepers that were trying to catch swarms, and you would have a tiny little swarm, like an after swarm almost, little bit of bees, tiny little comb on the swarm trap, and you could have 200 beetles waiting at the bottom for it to grow. It was incredible. So, but the numbers have gone down, and much like Lewis said, I've never seen any evidence that they hang around, and Hawaii would be the perfect place, rotten papayas, rotten pineapples, rotten wild guava, you name it. And some of the trials that we did in the lab, we were trying to offer fruits, and you really had to wait till the fruit was absolutely disgusting, completely spoiled, basically fermented, and before they actually fed on it. They were very, very reluctant to feed in anything that you could get off the market. So yeah, it was, like a truck full of fruits that you would consume would be not a good hospitable place for a small hive beetle.
Yeah, and yet they can find a bee colony a long ways
Oh yeah.
away.
Oh yeah. Oh yeah. They definitely can.
Heather had a question. What about using a felted woolen fabric instead of the artificial shop towel?
I can imagine Michaela. We tried. Michaela brought from Argentina where they actually have sheep, like the very, very nice wool, and we could not get the bees to do anything with it. We didn't try that hard. I agree that the woolen fabric would be good. But my understanding is the old-fashioned. First of all, it's super hard to find because I looked. The felt is now all synthetic pretty much, and the woolen one is really the one that I found was very expensive. So it's like, I don't know how good that would work. But again, and I don't know because again, we're in a subtropical climate and things work differently, and our beetles do peak and go up and down, but not probably like they do in the mainland with a more temperate, more seasonal environment. But it's amazing what can be done from the point of view of colonies with just simple management. Just most, against my advice, most of my beekeepers have dropped using any trap, and they do well. Some of the commercial guys in Big Island have also dropped using traps, and they do well. So I mean, you are going to have some losses. And one of the things that is really important, if you ever have a slime out, and if you, I will send Dewey a link to a video that we have on, oh, I think that one's in Spanish, but that's okay. You can get the gist. You can have horrendous slime out and dead colonies. Things that you never think a beetle would use, they will use. We had a beekeeper that had colonies. There was a flooding, and the little creek washed away like 50 colonies, and they were completely covered in mud, all dead. And he just took a couple days to pick up the equipment, and we were helping him pick him up. Inside, it was like a beetle fest. There were tremendous amounts. So you really need to be careful with things that you consider that they're unusable and dead, because you could have a big number. And in that case, we were scraping frames full of larvae. And what we did in that case was make big tubs of soapy water and just drown them, rather than using pesticides, rather than try to burn some of these things. It was just that. But again, because of environmental concerns and the closeness to the ocean, we didn't want to use any chemicals, except soapy water. But it was, you can really hurt yourself if you don't take care of your slime outs or colonies that are dead, or you leave materials around that can really recycle. And most of the problems that people seem to be experiencing in Latin America come from extraction moments, extraction houses, honey extraction times, rather than colonies themselves. So the beetles, can be controlled okay at the colony level, is when you have like a area where you bring lots of honey and you don't extract it right away, or you don't have a dehumidifier, because that's a really good way. If you have a dehumidifier that is not that expensive, a small container with a dehumidifier, you can put in frames with honey. You can crank that down. If you lower the humidity below 40% would do it, would kill the eggs in the very young larva. You can freeze them, but if you freeze them, you have to go for at least 2 days. So it's tricky. And that was one thing that really hurt the Hawaii beekeepers, is even with Varroa, we never had to worry about taking the supers. You could have ridiculous stacks throughout the year. And all of a sudden we were like, oh my God, because it was a space game. If you left things on top, you would basically be feeding beetles, things that were not patrol.
Yeah, and in the Pacific Northwest, it seems the honey house is an early warning for small hive beetles, particularly if you bring in any brood, some brood in those frames,
Yep.
which is frequently happens.
Yeah.
Our Carrie Yost is our Hawaii representative to the WAS board. And she put some interesting comments in our chat. Carrie, would you want to? I see you're unmuted. Would you want to say a little bit about what you're talking about with your treatments with small hive beetle?
Hi, this is Jim, Carrie's husband. Carrie's
Jim, I'm sorry.
out in Maui right now. So
okay, Jim.
Basically, we just have like a screen bottom board. It's like a drawer. It's like about 1x6 height, and I just make a drawer that slides into the bottom. Staple screen, the 1/8 inch best screen at the bottom, and take like a 11x12 bake pan or cookie muffin pan. Fill it with about a 1/2 inch of vegetable oil I get from like the local L&L fried food place, or
I know what that is.
I get like a 5-gallon jug of used oil for free, and that lasts me about a year. I put like a 1/2 inch layer of oil in these oil pans, slap it in this tray, and this tray slides in, the bottom board slides in as like a removable whole tray type thing in the bottom board, and the bees just pluck the beetles, drop them in there. So midweek, in between inspections, I can go out, pull a drawer out, see what kind of detritus is falling in there, be it large beetles, varroa, hive beetles, wax moths. I mean, I can just do a quick open the drawer, check, oh, nothing in there, close it back up. If I pull it out and see lots of junk, that's a sign I may have to take a look at that thing before my normal 2-week inspection. But, it's a real quick check for doing this. And I've had no problems with it in the last 8 years.
No, I agree that they do work. I'm going to share something, even though it wasn't supposed to be. Hang on, let me share this. Let me show you why I was okay. So you can see this other PowerPoint, right? So one of the things that we're working on and that we did, and it's like there are always beetles, in the bottom. It's not like they're not, but they, like when you check, when you move, when you disturb, or over time, they move more to the bottom. So one of the things is this that we did, and this is why I don't, I would prefer for people to sample on the top, because that's where they tend to go naturally by the behavior, than on the bottom. And yes, people have had screen bottom boards with deep things. There was even a company that was making these very, very deep pools. And you're good, Jim. You and Carrie are good because you go and you check your hives, but there are people who leave it forever, and when you do this, or when you don't check as much, or when you're not careful, the bees will propolize the screen. There'll be a reduced drop, or there'll be stuff sitting on top of the screen that can become a feeding area. So, but one bit of research that we did with Hawaii, Mexico, and Costa Rica was, and I took out the Mexico in this graph just to explain. One of the things that we did to test our idea was you lift a hive, closed hive, you haven't smoked it, you're nothing. You just go whoop and you lift the whole thing and you capture and kill whatever you have here. And what we found is this: Hawaii before, Costa Rica before. The number of beetles, the percentage of beetles that you would capture in a hive is really small when a hive hasn't been disturbed. When you go back and you work the hive, you put it back in a stand, you work the hive, you smoke it, you lift frames, you look at blah, blah, blah. Then you go back and you do the same thing. You lift the hive and you look at what now you have at the bottom. And you have a whole bunch of beetles that have moved to the bottom. So the bottom traps are really efficient for beekeepers that consistently work on their hives, that, in essence, scare the beetle into falling down. If you are not doing this very much and you want to take a quick peek, look at the top, because that's where most of them will be. So this is the case with Mexico, where we had colonies that had over 300 beetles in one instance. And there it was such chaos that you have beetles on top, beetles below, and there you had 30% of the beetles when the colony was addressed. Then when you collected beetles in here and you checked again, you had another 40%. So they did move down. It's just that there were so many there to begin with. And it also depends. In the case of Mexico, the hives, the bottom floors were made out of multiple pieces of wood glued together, so they're never exact, and there's always crevices, so there's always food, there's always stuff. But no, I agree with you guys. It can work with a bottom board. It's the amount of oil you need to use, the amount of mechanics you need to do is tricky. And these are some of the ones. This was used in Hilo when the small hive beetle first arrived, and these little dots here are beetles, and this is a one of those beetle barns, and you can see the outline where they had propolized the trap, glued it to the bottom, but also in reality they had glued the entrances as well, and you can see some dead beetles. And if you were to check for residues, there will be residues of fipronil where that thing was. And this is the stuff that I was mentioning. And the reason why we go with that is that it's a simple, and most beekeepers, if you're sampling your colony, you're going to open it anyway. If you're going to take a quick look and see how it's doing for honey. So you could potentially do this very easily. So that's why I was saying it.
Very good. Fascinating. Thank you, Jim, for sharing your method as well. Are there any other questions that individuals might have? Address? Well, not hearing any others, I wanted, and it's getting late, at least here on the East Coast for most of us. Thank you for joining and presenting this studies that are real. I found really fascinating, and I loved your presentation very much. And thank you to Michaela for joining us and joining in with you again this evening. Thank you. Thank you to Lewis.
Thank you so
Lewis
much.
was on for a while. He had to leave on the East Coast, but I'm sure it would be very beneficial for the two of you to try to have a discussion about East and West in terms of small hive beetles.
Yeah, tropical.
Yeah. Tropical.
And
Yeah, very
yeah.
good.
No, but thank you so much for the invite, and thanks for everybody. I know it's late, so many thanks, and have a good evening.
Thank you. Thank you all.
Okay.
Good evening,
Bye.
all. Bye-bye.
Good night.
Good night.